that they made those mutations. And more critical is the fact that these two insertions into the spike protein, which made spike proteins infective into humans above and beyond anything that naturally existed, produces changes in the rest of the molecule where the spike protein attaches to our cells. And that area is called the regional binding domain, and it's made changes that are called prion-like domains, which for the general public means they produce changes in the brain that produce the equivalent of mad cow disease or Alzheimer's disease. So this spike protein with these two insertions and then the subsequent change is a direct result of human intervention done following funding from the U.S. government for gain-of-function research that we shut down in the back almost a decade ago when the viruses that were the Asian flu viruses were being played with gain-of-function and got released accidentally, as well as some anthrax bacteria that had gain-of-function research being done to it, and the scientific community became alarmed. So this spike protein is the smoking gun that exposes the gain-of-function research that's been done to make a virus to be infective, not to kill people, but to carry out as a bioweapon to maim people and to harm economies and societies and shut them down. And that's exactly what's happened.
All right, Dr. Cahill, would you respond to that as well? Because I know that's something, again, we haven't talked very much about, but it really is alarming to think about.
Yeah, so I think when I came out as well, including on the High Wire in May 2020 with Dell, and in some of my interviews, there was a Nature paper in May 2020, and that described these 12 nucleotides, exactly the sequence, which is a furin receptor. And there was patents around it, as was just said. And in the patents, they said that it would increase the infectivity of any virus by 10 times to 1,000 times. So this gain-of-function type of research was made unlawful, including in the United States and in the European Union. So we now know that some of that research was carried on, was initially in America and then carried on in China. In a way, this was almost strategic, that China could blame America and that America could blame China, which is exactly true. But the reason why this man-made insert had to be put in is that they wanted to patent it or patent it. And there was rulings in America in 2011, 2012, that you couldn't actually patent or commercialize something that was just a natural virus. So that meant that was a big setback for the pharmaceutical and the diagnostic industry. So if they wanted to prepare to make the tests for COVID-19, as we know, many of them were ordered even as far back as 2017, 2018, they had to put some man-made element into it. They had to patent it, and that would allow them to make significant amounts of money and to prepare for what would be, look like it was an mRNA injection in the future, but that this would all have been patented and well described throughout this type of research involving, interacting networks between America and China and all around the world. So this gain-of-function is to make something man-made in order to patent it, so that then you could patent the PCR testing, the diagnostic testing, and any type of treatment.
So it's interesting, Adele.